fascinating world of organoids today.
Mm-hmm.
You've probably heard about these. They're the incredible miniature models of our organs that scientists can grow in the lab.
Yeah, really amazing tools.
We've seen them used for everything from like studying neurological disorders with brain organoids.
Gut issues, modeling viruses like SARS-CoV-2.
Right, and even heart organoids sent up to space, just to see how they handle microgravity. They're truly remarkable for exploring biology outside the body.
They offer powerful insights, definitely, into how organs develop and function, or, malfunction.
But there's always a but, isn't there?
Well, yeah. For years, they've run into this really fundamental limitation, a size problem.
Ah, okay.
No matter how sophisticated the lab techniques got, organoids just couldn't grow beyond a certain tiny size.
How tiny are we talking?
Roughly that of a sesame seed, about 3 millimeters in diameter.
The infamous sesame seed problem. And the source material explains why this limit has been such a persistent challenge, right?
Exactly. It comes down to the plumbing.
The plumbing.
Yeah, unlike actual organs in our bodies, traditional lab-grown organoids, they lack their own integrated blood vessel system.
So they don't have that built-in delivery network for essentials, like oxygen and nutrients.
Precisely. Those things can only reach the cells by diffusing in from the liquid they're grown in, just through the outer surface.
Okay.
So as the organoid gets larger, the cells in the center find themselves further and further away from this supply line.
They get starved, essentially.
Pretty much. As Oscar Abelez, one of the researchers at Stanford, puts it, he says, when you grow organoids to a certain size, they start to die inside because they can't get oxygen and nutrients to the center.
Wow.
Yeah. That lack of internal circulation has really been a bottleneck, limiting how large and, frankly, how complex these models can become.
But now there's this breakthrough from a team at Stanford Medicine that could fundamentally change all this.
That's the exciting part.
They figured out how to create the first heart and liver organoids that actually grow their own integrated blood vessels.
Yes. It feels like a huge leap forward.
It really does. This ability to spontaneously generate a vascular system, it directly addresses that core problem we were just talking about.
Right. It means these organoids have the potential not just to grow larger, escape that 3 millimeter constraint,
That also...
crucially, to reach a more mature and complex state, something that better mimics real tissue.
Okay, so our mission for this deep dive then is to really get into the details of this significant advance.
Let's do it.
We'll explore how this team, including Oscar Abales, Waxiao Adam Yang, and Joseph Wu, how they managed to crack the code and develop this recipe for vascularized organoids.
And
Then we'll dive into what this incredible step could mean for the future, both for medical research, like better modeling of disease and early development.
maybe even growing tissues for therapies.
Exactly. That's the long-term potential. We're pulling all the key insights directly from the Stanford Medicine source material here.
Sounds good.
So let's start by revisiting that core challenge again. Why was the lack of blood vessels the specific reason for that 3 millimeter limit?
Well, think about how vital blood flow is to every single cell in your body.
Right.
Your capillaries, those tiny vessels, they're everywhere. They deliver oxygen, nutrients, they remove waste products.
Constantly.
Without that intricate network inside the organoid itself, it's like trying to feed a huge crowd by only letting them get food from the very edges of a big room.
And the further into the crowd you go, the less likely you are to get fed.
Exactly. Diffusion, that passive movement of molecules, it works okay over tiny distances, maybe 1/10 of a millimeter efficiently.
But not much more.
No. Once the organoid diameter gets to around 3 millimeters, the cells just a millimeter or two inside are simply too far from the surface to survive.
That Abelez quote really nails it.
It does. When you grow organoids to a certain size, they start to die inside because they can't get oxygen and nutrients to the center. Overcoming this wasn't just like an engineering puzzle.
Right.
It was a fundamental biological one.
Which brings us to the Stanford solution, published in Science, no less.
Major journal. Yeah.
They didn't just make organoids. They made heart and liver organoids that could build their own internal plumbing system.
And this is the breakthrough that tackles that major bottleneck head on.
Right.
Integrated blood vessels just change the game. They offer a path for the organoids to potentially grow larger, function more like actual organs over time.
And beyond just being better research models, Joseph Wu, the senior author on the paper, he points to the potential in regenerative medicine, too.
Yes, he contrasts this with some current approaches. For instance, In a separate clinical study he leads, they're exploring injecting just one type of lab-grown cell,
Which type?
cardiomyocytes, the heart muscle cells, into patients with heart damage. But real heart tissue isn't just one cell type, is it? It's this complex mix. Cardiomyocytes, yes, but also the endothelial cells lining the vessels, smooth muscle cells around them, fibroblasts, immune cells. The list goes on.
So the long-term vision here is potentially growing something much more complex, like a miniature tissue patch with its own vessels grown from a patient's own cell.
That's the really exciting potential down the line, and the vascularization is absolutely critical for that to ever work.
Why critical?
As Abeles explains it, the thought is that if organoids have a vascular system, they could connect with the host vasculature, meaning the patient's own blood vessels after implanting.
Ah, so they could hook up, basically.
Exactly.
Yeah.
And that'll give them a better chance to survive.
Yeah.
An implanted piece of tissue without its own blood supply would face the exact same starvation problem the organoids had in the dish.
Okay, the what and the why are incredibly compelling. Now for the how. How did they actually pull this off? The source mentions previous attempts weren't great.
Right? People had tried. Traditionally, organoids are grown from these pluripotent stem cells. Yep. Cells that can become any cell type.
The master cells.
Kind of, yeah. Using chemical recipes.
Okay.
These are specific cocktails, mixtures of growth factors and small molecules that basically tell the stem cells what to become.
Okay.
Previous efforts to get vessels were often sort of hit or miss. They couldn't reliably get the right mix of cells needed. Or they involved trying to engineer vessels separately, maybe 3D printing structures, and then adding them to the organoid later.
And those engineered solutions just weren't quite hitting the mark. They weren't realistic enough.
According to Abeles, yeah, those methods often resulted in structures that don't really make branch vessels with passageways. They just weren't realistic, functional vessel networks.
So not like real capillaries.
Not really. So the Stanford team took a different approach. They focused on optimizing the original chemical recipe right from the very beginning.
How so?
Their goal was to reliably guide the stem cells to produce not just the main organ cells, like cardiomyocytes for the heart, but simultaneously the other cells essential for building blood vessels, the endothelial cells and the smooth muscle cells, all together in the same growing environment.
So they weren't trying to assemble pre-made parts. They were teaching the stem cells to build the whole thing, plumbing included internally, from scratch.
Exactly right. They reviewed existing methods for differentiating these three key cell types individually. Then, quite ingeniously, they combined elements of those methods into 34 different potential recipes or growing conditions.
34? Wow.
Yeah, each recipe varied the specific signals, the timing of adding chemicals.
How did they possibly sift through 34 different conditions to find the best one? That sounds like a lot of work.
They used a really clever tracking system. They modified the starting stem cells
In what way?
so that as the cells differentiated into those three key target types, cardiomyocytes, endothelial cells, smooth muscle cells, they would start to glow, fluoresce in different distinct colors under a microscope.
Ah. So the more successful a recipe was at making that desired mix of cells, the more colorful the resulting organoid would look. It was a literal visual readout of success.
Precisely. A multicolored indicator. They ran all 34 conditions for about 2 weeks and then imaged the organoids.
Yeah. Was there a winner?
Abeles said the results were pretty obvious. Condition 32 was the clear winner.
Condition 32.
Yep. It produced what they called the most colorful organoid, meaning it reliably generated the highest quantities of all three essential cell types needed for heart tissue with its vascular components.
Okay, so it glowed the brightest with all the right colors, but what did these winning organoids actually look like structurally? Was it just a blob of colored cells?
No, not at all. Under detailed 3D microscopy, they observed a very specific structure forming.
Like what?
The organoids were kind of donut-shaped. The cardiomyocytes and smooth muscle cells made up the bulk of the tissue inside.
And the vessel cells.
The endothelial cells formed an outer layer, and within that layer, and extending into the core of the organoid, they could see unmistakable blood vessels.
Really? Actual vessels?
Described as tiny, branching, tubular structures. They look like capillaries, ranging from about 10 to 100 microns in diameter.
So roughly the width of a human hair.
About that, yeah. So crucially, they weren't just random glowing cells. They were forming organized, branching vessel networks. Real plumbing.
That is truly remarkable. And wasn't there an unexpected bonus, too, in terms of complexity?
Yes, absolutely, using a technique called single-cell RNA sequencing.
Which tells you what types of cells are present.
Exactly. It identifies every different cell type there. They analyzed the cells in those winning condition 32 organoids, and they were genuinely surprised.
Why? What did they find?
They found that the organoids contain 15 to 17 different cell types in total.
15 to 17. Not just the three they aimed for.
Right. And this level of cellular diversity is incredibly close to what you'd find in a 6-week-old embryonic human heart, which has about 16 distinct cell types.
Wow, so it's like a snapshot of early heart development.
Pretty much. An adult heart is more complex, maybe 21 cell types. But getting 15 to 17 was, as Abeles called it, unexpected in a positive way. Their recipe, aimed at just 3 key cell types, ended up producing a much more complex and realistic tissue model than they initially anticipated.
Okay, let's pull all this together now. Why does this advance truly matter? What's the real impact here on research, maybe on therapy down the line?
Well, first, for research itself, the fact that this winning recipe seems to mimic the conditions of early embryonic development, that's huge.
Why specifically?
Because it means these vascularized organoids are incredibly valuable models for studying that critical period. The formation of different cell types, the early vasculature developing, that's often a black box in human development.
Because it's hard to study directly. Ethically complex.
Extremely challenging and ethically complex, yes. So these organoids open up a window, a way to look into a previously pretty inaccessible stage of our own development.
That makes sense?
And because they have the potential to grow larger and more mature now, thanks to their vessels, they become even better mirrors. Better tools for understanding both normal early development and maybe what goes wrong to cause congenital defects or diseases later in life.
And they've already used them to test a drug effect, right? Fentanyl.
They did, as a proof of principle. They exposed the vascularized heart organoids to fentanyl.
And what happened?
They observed a notable effect.
Yeah.
It actually led to the generation of more blood vessels within the organoid.
More vessels. Is that good or bad?
Well, that's the thing. The implications of that specific finding for, say, newborns exposed to fentanyl aren't fully understood yet. As Abeles notes, we don't know how that might manifest in a newborn yet, but it's a difference.
But the key point is the model worked. It showed an effect.
Exactly. It powerfully shows that these more complex organoids can pick up subtle biological effects of drugs. They could serve as a better platform for testing drug safety and efficacy earlier, maybe more accurately, before going into humans.
Okay, and looking ahead, that potential for regenerative therapies feels huge. We touched on implanting these.
Yes, the vision is definitely to move beyond just injecting single-cell types. While that strategy has shown some promise,
It's limited.
it's limited. The goal for really significant tissue repair, especially in complex organs like the heart, is to replace damaged tissue with something that replicates the natural structure and function as closely as possible.
Which means?
Multiple cell types, organized correctly and crucially with a functional blood supply built in.
So the idea would be grow a vascularized organoid from your own cells, maybe even fix a genetic issue in those cells first, and then implant that piece of engineered tissue.
That's the long-term hope. The key is that built-in vascular system. By having their own vessels ready to connect to anastomose with the patient's existing blood supply.
They're more likely to survive the transplant.
Much higher likelihood of surviving, yes. And integrating into the host tissue, providing real, long-term functional benefits compared to implants that don't have their own vascular network ready to go.
And this technique isn't limited to just heart tissue, which is also incredibly exciting. They tried it on liver, too.
Yes, they did, which shows it's potentially broadly applicable. The researchers successfully adapted this vascularization strategy to grow liver organoids.
And they also developed blood vessels.
They did. Robust, branching blood vessel networks. It involved optimizing a different recipe, obviously, for key liver cell types, but using the same underlying principle, co-differentiating the vessel cells right from the start.
So the core concept works for different organs.
It seems so. As Abeles says, he'd love to extend this. He said, I'd love to be able to do this in all the different organoid types. After all, almost every organ in our body has a blood vessel system. Makes sense, right?
Absolutely. So where do they go from here? What are the next steps with this breakthrough?
Well, the immediate next steps involve letting these vascularized organoids grow for longer periods. See how much larger they can actually get, how much more mature they can become over time now that they have supplies.
Pushing past that 3 millimeter limit.
Exactly. They're also working on refining the recipes further, trying to include even more of the cell types found in adult organs, maybe specific immune cells or types of blood
Just to
cell.
make them even better models.
Right, better models for studying adult onset diseases, not just development. And of course, applying this whole vascularization strategy to create more realistic, more functional organoid models for other organs throughout the body, lungs, kidneys, brain.
The possibilities seem enormous.
It really feels like this breakthrough, creating organoids with their own integrated blood vessels, is truly a transformative step in the field.
Yeah.
It potentially unlocks entirely new avenues for both understanding the just intricate processes of human development and disease, and for developing more effective, more tissue-like regenerative therapies for the future.
So for you, the listener, what this means is we're getting significantly closer to having laboratory models that behave much more like the real human tissues they represent.
Which is a big deal.
It could accelerate the discovery of new drugs, new treatments, by making testing faster, maybe safer, more accurate.
And it brings us a tangible step closer to that, well, extraordinary possibility of growing replacement tissues, maybe even mini organs, customized using your own cells one day.
Imagine that.
Consider for a moment the implications here. Scientists can now grow these miniature self-plumbing organs that give us a real window into early human development.
Or potentially grow functional pieces of tissue tailored just for you for medical repair down the road.
It really opens things up.
So what possibilities and maybe what profound questions does this remarkable step raise for you? As we look at the future of medicine and really our ability to mimic and potentially repair life itself.